ಇದು ವೆಬಿನಾರ್ಗೆ ಜಾಯ್ನ್ ಆಗಿರುವ ಎಲ್ಲ ವಿದ್ಯಾರ್ಥಿಗಳಿಗೂ ಸಹ ಸ್ವಾಗತವನ್ನು ಕೋರ್ತೀನಿ. ಇಂದು ಜೆನೆಟಿಕ್ ರಿಸೋರ್ಸಸ್ ಕನ್ಸರ್ವೇಷನ್ ಅಂಡ್ ಯೂಸ್ ಎಂಬ ವಿಷಯದ ಮೇಲೆ ಖ್ಯಾತ ಕೃಷಿ ತಜ್ಞರಾದ ಡಾಕ್ಟರ್ CLL ಗೌಡ ಸರ್ ಅವರು, ಫಾರ್ಮರ್ ಡೆಪ್ಟಿ ಡೈರೆಕ್ಟರ್ ಜನರಲ್ ಇಕ್ರಿಸಾಟ್ ಹೈದರಾಬಾದ್ ಅವರು ಹಾಗೂ ಕೋಫೌಂಡರ್ GRSV ಕನ್ಸಲ್ಟಿಂಗ್ ಸರ್ವಿಸಸ್ ಮೈಸೂರು ಸರ್ ಅವರು ಈಗ ಉಪನ್ಯಾಸವನ್ನು ನೀಡಿದ್ದಾರೆ. ಅವರ ಉಪನ್ಯಾಸಕ್ಕೆ ಮೊದಲು ಕಿರು ಪರಿಚಯವನ್ನು ಈ ಸಂದರ್ಭದಲ್ಲಿ ಮಾಡಿಕೊಡ್ಲಿಕ್ಕೆ ಡಾಕ್ಟರ್ CLL Gowda did his BSc Agri at College of Agriculture, Dharwad. Then pursued MSc from GB Pant University of Agriculture and Technology, Pantnagar, Uttarakhand. Then pursued PhD in agriculture from Indian Agricultural Research Institute, New Delhi, India. ಒಂದು ನಿಮಿಷ ಆಗಿದೆ.
ವಾಯ್ಸ್ ಬ್ರೇಕ್ ಆಗ್ತಾ ಇದೆ.
ಸಾರಿ.
ಆಗಿದೆ.
Sorry, sir.
No problem.
Okay, thank you. Dr. Laxmipathi Gowda sir, formerly the Deputy Director General, Kstacademy, Hyderabad. He earlier worked with Kstacademy as Director, Grain Legumes Research Program and Director of Crop Improvement Program. He was also the coordinator of Cereals and Legumes Asia Network during 1992 to 2014, and he is also worked with 12 Asian countries to strengthen the capacity of the national programs. He has also worked as a consultant to NARS, Bangladesh, Indonesia, Nepal, and India. As chickpea breeder, he has developed and assisted in the release of more than 70 hybrid chickpea varieties in 30 countries that have made significant impacts on farmers' yields. Dr. Gowda sir was one of the pioneers who initiated the public-private partnership for hybrid parents research consortium, ICRISAT, which is now serving as a model for other national programs in India and for CGIAR centers. Dr. Gowda sir has been honored and recognized by Bangladesh, China, India, Nepal, Philippines, Thailand, and Vietnam for his contribution to strengthen the national culture R&D. Has received 30 awards, honors, and recognitions for various countries, from various countries, institutions, and professional bodies across the world. More recently, he received Doreen National Award for Lifetime Achievement for Excellence in Science in 2012 and International Service in Crop Science Award in USA in 2012, the Japan International Food Award in 2014. He was also elected as a Fellow of the National Academy of Agricultural Sciences, India in 2015, a Fellow of American Society of Agronomy in 2015, Honorary Fellow of Karnataka Science Academy in 2020. He was also conferred the Karnataka Kalpamcha Award for his service to smallholders, farmers in the dry land areas, and was honored as a distinguished alumni by Alumni Association of Agricultural Sciences, Bangalore in November 2016, and by the University of Mysore Alumni Association in November 2021. Dr. Gowda has traveled widely, has 80 publications, including journal articles, 52 books, 54 book chapters, 30 research bulletins, manuals, and 52 conferences. His credit is also the chief editor of Asian Agri History Journal, editor of the Journal of Agriculture Research, so on, and International Journal of Production. Is currently one of the co-founders of the PRSC Research Service, which provides consultancy support to institutions and individuals in areas related to agriculture, research, development, including on-farm research, organic farming, research management, business, and public-private. Sir, it is my honor and privilege to welcome you once again to deliver a talk on genetic resources, conservation, and use. Sure, students will be highly benefited by your talk. Now, over to you for your presentation. Thank you.
Thank you, Dr. Ramesh, for that introduction. Good afternoon, everyone. Today's talk will be on genetic resources conservation and use, and I will possibly speak for about 30 minutes, and then if there are any questions or comments, queries, I can take those after the completion of the talk. The presentation will have the following topic that I would like to cover. First one is to indicate what is actually genetic resources, or also called as germplasm. What is the need for conserving these genetic resources, and how do we conserve these genetic resources? What are the global plant genetic resources that are currently under conservation and use? Different methods of germplasm conservation that are happening around the world. We also talk about biopiracy and related issues. We'll also briefly talk about the International Treaty on Plant Genetic Resources for Food and Agriculture, that is ITPGRFA, and then briefly also mention about the National Biodiversity Authority, or NBA, of India. Okay, coming to what is actually genetic resources or germplasm. Germplasm refers to seeds or plants or plant parts that are useful in crop breeding or crop improvement, in research, whether it is physiological research or other related research, and also its conservation efforts. Plants or seeds or tubers, rhizomes, cuttings, or cultures or germplasm, when they are maintained for the purpose of studying, managing, or using these genetic information that they possess. Together, they are called as germplasm. This germplasm actually is a live information source for all the genes that are present in the respective plants, which can be conserved for long periods and regenerated whenever it is required for the future. There are different classes of germplasm or genetic resources, and I have listed some of these. There could be land races, which are the varieties or lines that have been cultivated for a long time. There could also be obsolete cultivars or varieties. That means those cultivars or varieties that were being cultivated but may not be cultivated now. These could be modern cultivars or varieties that are currently under cultivation. These could be advanced breeding materials or breeding lines. There could be wild farms or races of cultivated species. There could be wild relatives of the cultivated species. There could be mutants or other special genetic stocks. Just to clarify, although germplasm is for both animals and plants, I would be speaking today mostly on plant genetic resources only. So, next question is, why do we need to conserve this genetic diversity? We need to collect and then conserve this diversity before it becomes extinct or gets eliminated from the earth. This loss of diversity or genetic erosion can happen due to the following reasons. One is deforestation and loss of habitat, urbanization and housing activities which take up land where possibly some of the wild species or some of the crop relatives would have been there in nature. Agriculture itself and its related activities can result in loss of genetic diversity. The fires that happen, both forest fires and bush fires, can lead to extinction of some of the diversity that exists globally. Flooding, another example. Loss of crop diversity in the farms, in the sense that in the earlier days, a farmer would possibly grow maybe 10, 12, 15 different crops in their farms. But however, now, because of specialization and other things, farmers are growing maybe 1 or 2 crops or maximum 3 crops. So this also leads to loss of diversity. And then the cultivation of uniform varieties such as hybrids or other highly uniform varieties can also lead to loss of genetic diversity. So this germplasm conservation, it involves different methods to conserve the genetic traits of endangered and commercially valuable species that we need to conserve for the future and also for use currently or in the future. Genetic diversity, therefore, is preserved in what we call gene banks. I will come to this a little later. And this conservation in the gene banks acts as a resource house for new or unidentified species. So gene banks are those structures where all the germplasm that is collected by an individual or an institution is stored under different conditions. I will come to the details a little later. The International Board for Plant Genetic Resources, IBPGR, was started by the Food and Agriculture Organization in 1974 as a global organization for germplasm conservation. IBPGR was renamed in 1994 as International Plant Genetic Resources Institute with its headquarters in Rome, Italy. And this was again renamed as Bioversity International in 2006. Currently, Bioversity and CIAT, the International Center for Tropical Agriculture based in Cali, Colombia, are jointly together. They call themselves as Alliance of CIAT and Bioversity International. Actually, Bioversity International provided essential support for collection, conservation, and utilization of plant genetic resources throughout the world. In addition to biodiversity, there is also another institution called the Global Crop Diversity Trust, or GCDT, which aims to conserve and make available germplasm to users globally. So in terms of conservation, what are the different steps? The first and foremost is to explore the centers of origin or centers of diversity and the hotspots where the crop is grown and the diversity is abundantly available. And then we plan the collection of germplasm from its habitats or in cultivated areas. And when we collect these germplasm, we also collect what we call the passport data for each sample. So this passport data actually consists of many different information that is necessary for future cataloging of the data. So essentially, it includes the area in which it was collected in terms of the location, the taluka or the district, the state, the country, and also it will have the geographic coordinates nowadays, where you can use the GPS and locate where exactly the germplasm was collected, and other data like the growing conditions, what was the condition of the crop, whether there were any pests or diseases, and similarly many other data are collected and cataloged, and a sample is collected and numbered according to an agreed standard process. So once these are collected, these are then grown out in adapted areas, because a sample would be maybe a few seeds, 10 seeds or 15 seeds or 20 seeds that would have been collected, but these may not be sufficient to keep in the gene bank, so they would be grown out and to harvest sufficient seeds, which we can then use it for storing. So once we harvest, we dry it, clean the harvested seed, and then we also test the seeds for seed moisture and germination. Seed moisture, because we should not have too much seed moisture, because high seed moisture cannot keep the seed for a longer time. So once we do the seed moisture test and germination test, then we keep the seeds in proper seed containers as per the storage needs. So in terms of methods of germplasm conservation, there are many methods. One is in situ conservation, where the particular crop species is conserved on site in the reserved biospheres or natural parks or sanctuaries where it is grown. Then the second one is what we call ex situ conservation, which is off-site conservation of the species outside its natural habitats, either as in vitro cultures or as seeds, which are then stored in the gene banks for long-term use. Third is cryopreservation of either cells or tissues at very low temperatures, with low temperatures and the deep freezers at -80 degrees, using vapor nitrogen at -150 or liquid nitrogen at -196 degrees Celsius. Then it's also cold storage, where it is conserved at non-freezing temperature between 1 to 9 degree Celsius. I will give a little more details when I go through next few slides. And then low pressure and low oxygen storage to reduce the availability of oxygen and less production of carbon dioxide, and hence photosynthetic activity is reduced so that the seeds can be stored for a longer time. So in terms of global germplasm available for crop improvement, as you can see here, globally there are about 7.4 million accessions of germplasm of different crops. On the left, you will see many crops like cereals consist about 45%, and then we have food legumes about 15%, forages 9%, vegetables 7%, nuts, fruits and berries for 6%. Similarly, other crops that are grown there. So these 74 lakh accessions are kept in 1,750, more than 1,750 gene banks. Out of these, people have estimated that only about 25 to 30% are unique. Some of them could be duplicates, but we don't want to throw the duplicates because over long periods of their growth and for many years, things may have changed and they may have useful traits that may not be present in the original stocks. Out of these 74 lakhs, only 10% are crop relatives, wild relatives of the crops, and 11% of these, that means about 7 lakhs and odd, are stored in the CGIAR gene banks. So these are the CGIAR centers, what we call Consultative Group on International Agriculture Research, and there are currently 15 of these centers globally, but out of these 15, only 11 have gene banks or where they store the genetic resources. So these are CIMMYT, which for maize and wheat, CIAT, SIP for potato, IITA, WARDA, mostly for rice, IPGRI, I mentioned International Plant Genetic Resources Institute, which currently has arrangements with other institutions for storing banana, ICARDA, which was there in Aleppo, but currently now located in Morocco, ICRISAT in India, then we have ICRAF, we have CIFOR, and then IRRI for rice germplasm. There are many germplasm institutions in many countries. I have only listed a few of them here. In India, we have the National Bureau for Plant Genetic Resources based in New Delhi. In the USA, they have 2 or 3 gene banks. One of them is the National Plant Germplasm Program. In Russia, we have the Vavilov Institute for Plants. Japan has the National Institute for Agriculture Research. Ethiopia has the Plant Genetic Resources Research Center. China has the National Gene Bank located in Beijing. Portugal has its own gene bank. Sweden also has a gene bank. And as I mentioned, the CGIAR has 11 gene banks globally. So these are the major gene banks that are there. I have listed a few, but since the table is big, it is not possible for you to read. Just to indicate, India has about 4 lakh, 44 lakh 64,000 accessions. China has about 41 lakhs. So similarly, there are many other centers which have large collections of germplasm across. A bit more detail on germplasm conservation. I mentioned about the gene banks. One of them is what we call the seed gene banks, where seeds are kept. If it is what we call active collection, for where we can store the seed for 20 to 25 years, seeds are stored at 4 degrees Celsius, and the atmosphere in which the room in which it is stored should not have more than 30% relative humidity. With that, we can have seed viability of more than 85%, 80% for groundnut seeds and 85% for other crops. You may ask why 80% for groundnut seeds, because groundnut is an oilseed crop, and because of the oil content, the storability is slightly reduced for oilseed crops. For the base collection, where we can store the seed for more than 50 years, it is stored at minus 20 degrees with 3 to 7% seed moisture. So we have to dry the seeds to such an extent that the seed moisture comes down to between 3 and 7%. And in this, seed viability is kept more than 85% for groundnut and more than 90% for other crops. The first one is the seed banks, and then the second one is the field gene banks, where it is used for recalcitrant seeds, where it is difficult to grow the seeds, and also for tree species where we cannot store these in the gene banks. So it is there in the field gene banks, and then they are maintained throughout their life cycle. We also have in vitro gene banks for vegetatively propagated crops like potato, sweet potato, and many others, where in vitro conservation under slow growth in active gene banks, and also cryopreservation in with very low temperatures. This is one of the things that the Global Crop Diversity Trust and the Biodiversity International have done in terms of trying to keep a safety duplicate of all the germplasm in the world in another site. So should something happen in one country, let us say war happens and then a bomb falls on the gene bank, the national gene bank, the whole thing is lost. So for that, it's always advisable to have a safety duplication in another place, possibly in another country. So this is one of those global initiatives where safety duplication is there in the Svalbard Global Seed Vault, which was it is established on the Norwegian island of Spitsbergen in the Arctic Svalbard archipelago. And here, more than 1 million, about 10.5 lakh samples, major crops of both wheat, rice, maize, coffee, sorghum, and also many other crops are stored. The CJR gene banks, the 11 gene banks that I mentioned, have deposited most of their germplasm, duplicate samples of their germplasm samples in the Svalbard gene bank, Global Seed Vault in Norway. Just to give an example of how the CJR gene banks keep their material, this is an example from ICRISAT, where currently they have about 1,24,000 germplasm accessions. Out of these, active collections are about 1,24,000, and then the base collection is about 1,11,000. So active collection are those where these are regenerated and are available for people to access them. Base collections are those where they are kept in those minus 20 degrees for long-term conservation. Just a brief look on the different diversity that is there so that people can understand what exactly diversity means. This is the sorghum diversity for earhead size, shape, and seed color, and things like that. Just a few examples. There are many more diversity in the global collection, but I just gave a few examples here. This is the diversity of pearl millet earhead. You can look at the smallest size is about 1 inch, and then the longest would be possibly about 1 meter. And then you have things that are awnless. Then there are earheads with awns. Similarly, there are many other traits like seed color, seed size, shape, and many other diversity. This is the diversity for chickpea germplasm seeds. Again, you can see different varied colors, shape, and size. This is the diversity for pigeon pea or red gram germplasm seeds. Then the diversity for groundnut germplasm seeds as well as pods that are available in the ICRISAT germplasm. So I would just like to briefly talk about biopiracy of genetic resources, which means a wrongful appropriation. That is, somebody picking up the genetic resources of one country, taking it elsewhere, and then taking an intellectual property right or patenting on that biological entity or the active agent derived from that particular entity or to the knowledge associated with that particular genetic resources. So, although developed countries receive extensive support on intellectual property decisions, low-income countries are unaware of what patent is and how to manage these patents. So therefore, the International Treaty on Plant Genetic Resources for Food and Agriculture, that is ITPGRFA, plans to protect the germplasm of each country and the rights of each country for equitable sharing of the benefits of these plant genetic resources. But unfortunately, very few developed countries have ratified this treaty for their own reasons. You will understand the political reasons why they would not like to ratify the treaty. Examples of biopiracy, for example, the basmati rice from India and Pakistan, jasmine rice from Thailand, several medicinal and aromatic plants and products from India. Major ones are ginger and many others, which people from other countries have tried to claim patent on. So how to minimize biopiracy is we need participation of everybody, both rich and poor countries, in the FAO's international treaty, and its effective implementation for equity across all the countries. So we need to have documentation of the traditional knowledge in the digital form and linking it to the internationally accepted patent classification system so that if somebody else wants to apply for a patent, that information and data is available so that they can say, look, this information already exists in this particular country, so you cannot take a patent on this. So I already mentioned about the International Treaty on Plant Genetic Resources. 131 countries are parties to the treaty, including India. India has ratified the treaty. All CJR centers are party to the treaty, and this ITPGRFA has put up what they call a Standard Material Transfer Agreement, SMTA, which has been implemented. And this Standard Material Transfer Agreement is the one through which people can exchange material from one place to another, from one institution to another institution. So if somebody asks a CJR center for germplasm, then the person who is asking for that needs to sign this SMTA agreeing to certain conditions for the other party to send that material. So this needs to be reported to the governing body, which is the FAO. And in this multilateral system of ITPGRFA, there is what they call Annexure 1, which has 35 crops which are in the multilateral system, which means they can be exchanged among different countries or different institutions, plus 79 forages. Not all crops are included, just note that. Some countries have refused to put their material in Annexure 1. For example, Malaysia has not put oil palm in the multilateral system because they wanted to protect the oil palm industry in their own country. Sorry, this seems to be a problem. Slides are not moving. Okay, yeah. Seems to be moving now. So in the ITPGRFA, the Article 5 says each contracting party should promote an integrated approach to the exploration, conservation, and sustainable use of plant genetic resources for food and agriculture. So these are the objectives in terms of conservation and sustainable use, and then the fair and equitable sharing of genetic resources derived from their use for agriculture and food security. And Article 12.4 of this treaty specify the access of the plant genetic resources through use of SMTA. I mentioned about the Standard Material Transfer Agreement. And then this SMTA has terms and conditions that cannot be changed by anybody. So this implementation of SMTA is the agreement between the provider and the recipient, that is the SMTA, and treaties, objectives, and legal measures to be followed. And the plant genetic resources should be used or conserved only for research, breeding, and training for food and agriculture. This does not apply for chemical, pharma, and other non-food and feed or industrial uses. For that, there are other methods through which countries can exchange the genetic resources. So these are the rights and obligations of the provider. Access shall be accorded along with associated information. Usually it should be given free, but they can put a fee based on the minimum cost that is needed to produce the seed, and then needs to report it to the governing body about the SMTAs entered yearly. Recipients' rights and obligations, they are not allowed to take a patent protection on material per se, or its genetic parts, or information associated with it. They are authorized to make third-party transfers under the new MTA with similar terms, and then they should notify all third-party transfer to the governing board of the FAO. So no more obligations beyond that. So if we talk about the National Biodiversity Act, its implication for research institutions, there are no restrictions from the NBA, National Biodiversity Authority, for receiving any Indian biological resources for research use with SMTA. They can also share the research on any Indian biological resource to any other recipient within India. They can share the Indian biological resources to any other recipient within India with SMTA, and sharing of the designated germplasm to non-Indian sources in India and abroad. They need to obtain the approval from the National Biodiversity Authority. So NBA's approval is required for transfer of any Indian biological resource to any person or any purpose outside India, and the transfer of results of any research on Indian biological resources also, if it is shared outside India, needs to be approved by NBA, and similarly any other biological resource to other locations. To conclude, all we can say is crop germplasm are basic for crop improvement for sustainable agriculture. We also know that all the germplasm is not used, so we need to enhance the use of germplasm. We also have small-sized subsets that have been formed for identification of new and diverse sources. Wild relatives possess novel alleles for developing broad-based cultivars, and we need to be aware of the National Biodiversity Authority and the ITPGRFA regulations for exchange of germplasm. So with that, I'd like to thank you for your attention, and then I will hand it over to Dr. Ramesh for any questions or comments. Thank you.
Thank you very much for a very informative and insightful talk on genetic resources, conservation and use. We spoke on genetic resources to conserve the ensure students who have got lot of message from your talk. On behalf of KSTA, all the partners and investors, thankful to you, sir. Umesh Gatkar, are there any questions? Sir, question.
Yeah.
In Norway, it seems they are preserving some grains and seeds for future. If there is any problem.
Right.
Like to
This is what we call safety duplication of the germplasm. So in Norway, in the one of the islands which is near to the Arctic area, where you don't need to cool the temperatures artificially, because it is already cold, because it is almost 10 months of the year or 9 months of the year, it is covered with ice. So there they have put up this global seed vault, and it is stored there with minimal cost, so that if something happens to the national or international collection, they can always go back and ask the global seed vault to give their germplasm back, so that they can grow out and re-establish their gene bank.
thank you very much, sir, for a very enlightening. As of now, If students ask, we will be posting the question to you, sir. I request you to answer if students mail.
Yeah. Okay, so I think they would have put up their emails so I can respond to them.
Yes, sir. Yes, sir. They will
Okay.
post to us.
Hmm.
We will pay the same to you, sir.
Okay, okay. Right. Yeah, that's okay.
ಎಲ್ಲ ವಿದ್ಯಾರ್ಥಿಗಳು ಸರ್ ಇವತ್ತು ಬಹಳ ಉತ್ತಮವಾದ ಮಾಹಿತಿಯನ್ನು ನಮ್ಗೆಲ್ಲರಿಗೂ ನೀಡಿದ್ದಾರೆ. ಎಲ್ಲರ ಪರವಾಗಿ ಸರ್ ಅವರಿಗೆ ಮತ್ತೊಮ್ಮೆ ಧನ್ಯವಾದಗಳು. ಮತ್ತೆ ಸೋಮವಾರ ಇನ್ನೊಂದು ವಿಷಯದ ಬಗ್ಗೆ ತಜ್ಞರು ಉಪನ್ಯಾಸ ನೀಡ್ತಾರೆ. ಇದೇ ಸಮಯಕ್ಕೆ 4 ರಿಂದ 5 ನಾವು ಜಾಯ್ನ್ ಆಗಬೇಕು ಅಂತ ಈ ಸಂದರ್ಭದಲ್ಲಿ ಕೋರ್ಕೊಳ್ತೀನಿ. ಇವರಿಗೆ ಧನ್ಯವಾದ. ಥ್ಯಾಂಕ್ ಯು.
Thank you.